The overall goal of this procedure is to derive a population of purified neuronal progenitors from peripheral nervous system ganglia. This is accomplished by first dissecting the organ or specific ganglia of interest from fetal mice. The tissues are then partially dissociated to free the individual cells into the surrounding media. Next, the cell suspension is filtered to remove any residual tissue clumps or large aggregates. Finally, the cell suspension is flow sorted to isolate the population of interest based on the expression of a fluorescent reporter in that cell population. Ultimately, gene expression patterns can be compared between progenitors isolated at different stages of development, or between purified populations of different neuronal cell types.
The main advantage of this technique over isolation of cell populations based on antibody labeling is that often immunoreagents are not available that label cells expressing a particular gene. Genetically engineered lines of mice that produce fluorescent protein in discrete neuronal subsets enable isolation of these cell populations.
While we demonstrate this method for isolation of neural progenitors from intestine and urogenital tract of the mouse, it can also be applied to other model systems that express fluorescent reporters.
Generally, individuals new to this method will struggle because they tend to overdigest the source tissues, resulting in a loss of cell viability.